The brain is one of the most enigmatic and yet amazing organs to exist, ever. Without the brain, your body is just a bunch of parts. What your nervous system does is bring all of these together to get life in motion. All in all, it's thanks to our good friend up here that we can move, speak, smell, feel, and pretty much live. And living is kind of important to lots of people, including myself. So understanding how the brain works can not only give us a better appreciation of life, but also allow us to make life better by responding directly to our brain's needs. Except it's not that easy. In fact, it can be really, really hard. Unlike IKEA, the brain is not compartmentalized into specific sections. Like IKEA, however, the brain is a maze. It's not like we have hunger here, left leg motion over there, and a sense of humor in the middle. It's more like there's a bit of everything everywhere. And when I say everywhere, I mean every single spot. And when I say a bit of everything, I mean more things than we can possibly ever imagine. Thus, at the end of the day, studying the brain can feel like a shot in the dark. But what if there could be light? I present you Optogenetics.
Ba-ba-ba-da-boop. Ah!
Optogenetics is a technique which gives us the capacity to control cell behavior by turning on a light. How exactly? Asks the intrigued and curious thinker at the back of the room. Well, intrigued and curious thinker at the back of the room, let's take a step back for a minute. It's time for you to meet someone. This is a pond algae. More specifically, this unicellular organism is called Chlamydomonas. In order to survive, Chlamydomonas needs light for photosynthesis. It swims towards the light, but it doesn't have eyes like us to see. So, how does our blind hero find its path? The answer lies in its eye spot, a small organelle filled with a specific opsin group. Opsin are light-sensitive proteins. In this case, we are dealing with an opsin called channel rhodopsin. When it receives blue light, this protein moves positively charged ions across the eye spot membrane, changing its voltage. If this change in voltage would be applied to electrically excitable neurons, the fundamental units of the nervous system, we could use channel rhodopsin to make specific neutrons active or even inactive. Surprisingly enough, most of our questions about life can be answered by life itself. We just have to listen. So, how does this relate back to optogenetics? Well, remember the part where I was like, what if we could apply this algae light-sensitive protein to neurons? Yeah, spoiler alert, we can do that! In fact, that is precisely what optogenetics involves.
Step 1.
Build a construct! Take your opsin gene, the instructions on how to build your opsin, and bind it to a promoter. A DNA sequence that tells specific cells to read the instructions and carry them out.
Step 2.
Put your construct into a virus, a small infectious agent that replicates itself within cells.
Step 3.
Inject the virus into an animal brain. Common testing takes place with rats. The virus will only act on targeted neurons without harming the animal.
Step 4.
Insert an optrode into the animal brain. An optrode is an optical sensor device.
Step 5.
You've got your optrode in. Now, you can laser lights of specific wavelengths into the brain to open neuronal ion channels. Bada bing, bada boom, you are set up to study the brain. Or, really, a specific neuron in the brain. You can monitor changes in the voltage through electrodes and correlate these to behavior testings to see what they do. So what? How is this any different from before? Oh, you dashing thinker at the back of the room, I'm glad you asked. With the ability to selectively turn neurons on and off without affecting the other parts of the brain, we can finally understand what the neuron does and why it's important by observing the changes between its presence and its absence. So, this is really cool and all, but what can you do with this? Optogenetics can help many people with disorders rooted in neurological problems. Take depression, for example. Because some cells don't work properly, brain activity dies out earlier and doesn't reach the extent it should, the probable cause for the symptoms experienced. Antidepressants allow the activity to go further, but as medications, these chemicals don't just affect the cells that don't work. They affect everything, leading to side effects and really treating but not curing the problem. However, through optogenetics, because of its specificity, we can determine what cells need to be further activated and help them reach further activity levels through the use of lights. This can be applied universally to addiction, sleep depression, Parkinson's disease, epilepsy, and the list goes on and on and on. As Ed Boyden, neuroengineer at the MIT Media Lab, put it, these disorders do not only steal our time to live, but they also change our identity, our emotions, and who we are as people. So really, studying the brain goes beyond the pleasure of understanding and learning, and reaches the extent of turning what now seems like a shot in the dark into a second shot at a healthy and, moreover, happy life. Well, thoughtful thinker at the back of the room, we've come a long way, you and I. We're practically optogenetic experts by now, am I right? No, I'm not. There is so much more to learn, which is really awesome. Perhaps our brains are so complicated that we will never truly even have the possibility of being optogenetic experts. But we will always be thinkers, and for that, I think and feel eternally grateful.
Ba 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 da boop, ba ba da ba 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 da boop. Hey hey.